हेलो फ्रेंड्स, वेलकम बैक टू माय यूट्यूब चैनल। इस वीडियो में हम एमएससी जूलॉजी 4th सेमेस्टर सब्जेक्ट बायोकेमिस्ट्री का टॉपिक ऑक्सीडेटिव फास्फोराइलेशंस का स्टडी करेंगे, जिसका इंट्रोडक्शन है। ऑक्सीडेटिव फास्फोराइलेशंस इज ए क्रूशियल बायोकेमिकल प्रोसेस इन लिविंग ऑर्गेनिज्म रिस्पॉन्सिबल फॉर द एटीपी प्रोडक्शंस ऑफ एडेनोसिन ट्राइफॉस्फेट, द एनर्जी करेंसी ऑफ द सेल्स। This process occurs in the mitochondria of eukaryotic cells, and its significance lies in powering various cellular activities, including muscle contraction, nerve impulse transmissions, and biosynthesis. Understanding oxidative phosphorylation provides insight into how organisms convert in the energy stored in food molecules into a usable form, ensuring survival and functionality. Definitions. Oxidative phosphorylation is the metabolic pathway through which cells produce ATP by transferring electrons from electron donors such as NADH and FADH to oxygen via a series of protein complexes located in the inner mitochondrial membranes. This process couples electron transfer with the phosphorylation of ADP to ATP. Components of oxidative phosphorylation. First, electron transport chain. The ETC comprises 4 protein complexes, complex 1 to complex 4, embedded in the inner mitochondrial membranes. First, complex 1, NADH dehydrogenase, transfers electron from NADH to ubiquinone coenzyme Q. Complex 2, succinate dehydrogenase, transfers electron from FADH to ubiquinone. Complex 3, cytochrome BC1 complexes, transfers electron from reduced ubiquinone to cytochrome C. Complex 4, cytochrome C oxidase, transfers electron from cytochrome to C to molecular oxygen, reducing it to water. Second, electron carriers. Ubiquinone coenzyme Q, a lipid soluble molecule that transports electron between complex 1 and 2 and 3. Cytochrome C, a small water soluble protein that shuttles electron between complexes 3 and 4. Third, ATP synthesis. This enzyme uses the proton gradient generated by the ETC to synthesize ATP from ADP and inorganic phosphate. Mechanism of oxidative phosphorylation. First, electron transfers. Electrons from NADH and FADH generated during glycolysis, the Krebs cycle, and beta oxidations are passed through the ETC. Each transfer releases energy used to pump protons across the inner mitochondrial membranes from the matrix to intermembrane space. Second, proton gradient formation. The proton pumping creates an electrochemical gradient known as proton motive force (MPF), characterized by a high concentration of the protons in the intermembrane space and a lower concentrations in the mitochondrial matrix. Third, ATP synthesis. Proton flow back into the matrix through ATP synthesis, driven by the PMF. The energy released during this flow powers the conversions of ADP and PI into ATP. Fourth, oxygen as the final electron acceptor. At complex 4, oxygen accepts electron and combine with proton to form water, completing the processes. Significance of oxidative phosphorylation. First, ATP productions. The primary role of oxidative phosphorylation is to produce ATP, supplying energy for cellular process. Second, metabolic efficiency. Oxidative phosphorylation is highly efficient, generating approximately 34 ATP molecules from one glucose molecule. Third, cell survival. The process is vital for cells with high energy demands such as neurons and muscle cells. 4. Thermoregulations. Heat generated during oxidative phosphorylation help maintain body temperature in endothermic organisms. Regulations of oxidative phosphorylation. The process is tightly regulated to ensure energy production matches cellular needs. Key regulatory mechanisms include: first, availability of substrate. NADH, FADH, ADP and oxygen levels influence the rate of ATP synthesis. Second, ATP and ADP ratio. The high ATP levels inhibits the process while high ADP levels stimulate it. Third, feedback inhibitions. Products like ATP and NADH inhibits enzyme in the Krebs cycle, indirectly affecting oxidative phosphorylation. Disorder associated with oxidative phosphorylation. Defects in oxidative phosphorylation can lead to various mitochondrial diseases, including: first, like syndrome, a neurological disorders caused by mutation in ETC genes. Second, mitochondrial myopathies, muscles disorder resulting from impaired ATP productions. Third, Parkinson disease associated with dysfunction in complex 1 of the ETC. Experimental approaches to study oxidative phosphorylation. Respirometry measures oxygen consumption to assess mitochondrial function. Second, mitochondrial isolation enable the study of mitochondria component and ATP synthesis in vitro. Third, genetic studies identifies mutation affecting oxidative phosphorylation. Conclusion: oxidative phosphorylation is a fundamental process of cellular energy production, converting the energy from nutrients into ATP through a series of well-coordinated steps. Its efficiency, regulation, and integration with other metabolic pathways highlight its importance for life. Understanding this process has implications for addressing metabolic disorders, improving health, and developing therapeutic interventions. References: